Hello, everyone. What we talked about in the previous lecture was the thermodynamics behind the processes in living organisms, particularly in reactions. What we're going to talk about in this lecture is how do these reactions take place in living organisms under the control of these molecules called enzymes. Now, what we'll talk about first of all is why do we need enzymes? Why do living organisms need enzymes in order to control the reactions? And then we'll talk about how do enzymes take control of the reaction? How do they speed them up? And finally, how we'll talk about how the enzymes can be regulated, what parameters affect the activity of the enzyme, and which ones are used by cells or living organisms in order to control the activity of the enzyme. So the first question is, why do we need enzymes? If you recall, when we talked about Earth as a great place for life to start, we talked about the fact that how the distance from the sun allow Earth to have temperatures which keep water mostly in the liquid form. And because of this, create stability, and so life could evolve here on Earth. Now, that's great for stability. However, when it comes to reaction, that is actually a problem. Reaction in general, so chemicals, in order to interact with each other, they need usually high temperature. And so the reaction needs high temperature. The temperature here on Earth are not high enough for this reaction to happen in a significantly fast way, or most of the reaction. And so that's the first reason why we need enzymes. Without enzymes, living organisms could not perform the necessary reaction with enough speed compatible with life. The second thing is now that you have this enzyme recording or helping with the reactions, at the same time, also you have the advantage of having something that you can control. So you have actually a mechanism to turn this reaction on and off. So having enzymes controlling the reaction give living organism a way to control also when to produce something and when to turn off the production of the same molecule. So allow for the control of the reactions themselves as well. So obviously all life relies on enzymes. You know, there are hundreds of enzymes there. And what they do, they speed up the rates of the reaction. We'll talk about later on how do they do that. And to give you an idea how much they can speed up the reaction, is not always like this, but look at the example here with phosphatases. You remember these are those enzymes that are that what they do, they remove phosphate groups from molecules such as enzymes. You remember kinase, they add phosphate groups, phosphatases, they remove them. And just to give you an idea how fast they will work without enzymes, it would take about 1 trillion years. That's a big number for one molecule to be dephosphorylated. So to have that phosphate group removed. Plus, when you add instead the phosphatase, this happen like so much faster in 10 milliseconds. So we're talking about 10 to the 21st time faster than the reaction itself. So you can see how much enzyme can really help with this reaction, how much they can speed them up. Now let's focus on how do enzymes do that. So when we talk about the reaction and the thermodynamics of reaction, we talk about how through reaction you have a change in the free energy, free energy. Remember, which is dictated by changes in the enthalpy and entropy of the molecules involved in the reactions. And so what you have here, in reality, this is not really a reaction, but this is an entire process where you have glucose broken down into water and carbon dioxide. So this corresponds to the whole cellular respiration. But imagine this as a reaction. So what happens is that you have the molecules that are the reactants, and then they're converted into products with a release in energy in this case. So this is an exergonic process. But before this process takes place, or any reaction takes place, the reactants have to be first brought to a certain level of energy higher than the one. The difference between the level of energy where they have to be brought to, which corresponds to what is called the transition states of the reactants, the difference between that energy and the original energy of the reactants is referred to as the activation energy. Once the molecules reach this level of energy, then they can finally be converted into products. So this is needed in all reactions. The activation energy can be of different magnitudes, bigger, smaller, depending on the reaction itself. But it's necessary for all the reactions. Now, what do enzymes do? They act like biological catalysts. Whenever you talk about a catalyst, you'll see, particularly in chemistry, you're talking about some molecule that can speed up the rate of a reaction. So basically, how fast a reaction can go. And this can be a chemical or can be biological molecule. So enzymes are biological catalysts. They are biological molecules, organic molecules that do affect the rate of a reaction. They speed it up. Okay. And how do they do that? Is by lowering the activation energy. So that energy necessary to bring their reagents, in this case we call substrate when it comes to an enzymatic reaction, to that transition state. So now, enzymes, that sometimes they need additional molecules, they're called cofactors, and these can be either inorganic molecules, such as ions, for example, and this we refer to as pure cofactors, or when they are organic molecules, you see when we talk about cellular respiration and photosynthesis, we'll talk about NADH, FAH2. In this case, these are modified actually nucleotides, for example. They're organic molecules and they're referred to as coenzymes. So these are molecules that are not part of the protein, but they combine with the protein, and they are necessary for this protein, this enzyme, to perform its action. Now, as we said before, so enzymes work by reducing the activation energy. So this energy necessary for the reactants to reach the transition state and then being converted in product is greatly reduced. So just by doing that, you can speed up the reaction. Obviously, the chance of this molecule to reach the transition state is much higher than original, than without the enzyme. And so by doing that, you speed up overall reaction per se. Now, notice one thing. Enzymes, they reduce the activation energy, but they don't change at all the change in free energy of the reaction. So the delta G of a reaction is absolutely untouched by the enzymes. So, which means that for a reaction to happen, whether there is an enzyme or not, you remember it has to be exergonic. It will stay exergonic. It has to be exergonic even without the enzyme in order to happen. Okay? They don't change, don't make a spontaneous reaction non-spontaneous or vice versa, non-spontaneous reaction spontaneous. They don't affect the delta G. So if a reaction has to be spontaneous on its own in order for it to happen, or if you remember, it has to be combined, the spontaneous exergonic reaction with the energy coupling, so the breakdown of ATP in order to be made exergonic. So, but what they act on is just the activation energy. They just lower the activation energy there. How do they do that? Well, they do that by binding reactants, which in a reaction, in an enzymatic reactions are referred to as substrate. They bind it in a specific region of the protein. This will be produced by the tertiary or quaternary structure of the protein, which is referred to as the active site. The active site is that region where the substrate is bound to. And it's through this binding, and now this molecule can later on be converted into the product. Now, how do they bind it? There are different models here. The mostly accepted one is what is referred to as the induced fit model. And through the induced fit model, basically what it means is that the substrate is bound by the active site, immediately having almost a perfect fit to the substrate, but then the perfect fit is derived just after the binding itself. And so by doing that, it's the enzyme adapts, basically the active site adapt to the substrate, and by doing that, it's then ready to convert the substrate into the product itself. The enzyme, notice one thing, it binds to the substrate, converts it into the product, and then it stays unchanged. It's not a react. Okay, it just helps the reaction, but it stays unchanged through the overall process. Now, again, this shows you the cycle that you have with any enzyme. Here you have an example of beta-galactosidase, which is that enzyme that breaks down lactose into galactose and glucose. We talked about this disaccharide when we talked about carbohydrates. And so the way it works, the substrate is bound by the active site, and this is where then it gets basically converted into reactants, and then there's products. Then the products are released, and the enzyme is released unchanged and goes back to do the same reaction again on cycle. So you can see how the same enzyme molecules can perform many reactions on its own. So how does this translate into the reduction of the activation energy? Now, this is done through three possible mechanisms. So first one, imagine that you have two reactants, two molecules that have joined together. Well, you can reduce the activation energy simply by binding both of them and bringing them close to each other. If you think about that, if you have two molecules that have to be combined together, now, if they're free in a solution without an enzyme, you rely just on chance for these two molecules to meet each other and react with each other. Now, by having a common active site, these two molecules now, they're simply brought close to each other. You increase the chance of these two molecules reacting with each other, obviously if they're brought close to each other. The second thing is mechanic way. There's an induced fit model, right, that we're saying. So if you have a substrate, it's bound by the active site. As soon as the active site basically adapts to the substrate, it might cause in some way, so this will be this model right here, it might cause some strain to the molecule itself. And by doing that, it might make it more prone to, for example, be separated into two different molecules. So just a mechanical way to do that. The last thing is by having an interaction. So basically by bringing the substrate inside the active site, where you may have different R groups, not only ionic ones, but you can have just the polarity here itself, might cause an alteration in the substrate, which makes it more prone to either react or be separated into molecules. So all of these three mechanisms are possible, and all of them will result in the reduction of the activation energy of the reaction. And the last part, what we're going to talk about is the mechanism through which the activity of the enzyme can be regulated. So what factors can affect the activity of an enzyme, and how can they be used by cell to control ultimately the speed of a reaction? So there are different factors that can affect the activity of an enzyme, and therefore how fast the reaction, enzymatic reaction, can take place. Can it be slowed down or accelerated or stopped and so on? First factors is concentration of the enzyme itself and the concentration of the substrate. So imagine you have molecule A converted into molecule B, and this reaction is controlled by an enzyme E. Now, the concentration of the substrate A affects how fast the reaction can go. Same thing, the concentration of the enzyme itself. Together with that, there are a series of regulatory molecules in living organisms, that are organic regulatory molecules usually, that do affect the activity of the enzyme. That can be competitive, non-competitive. We'll talk about that later on. And then enzymes can be modified covalently. We talked about that when we talk about kinases in signal transduction. Finite temperature and pH has an effect on the enzyme. So let's go through all these factors here. First thing, concentration of enzymes. Now, assume you're in a situation, you have a reaction, A converted into B, under the control of this enzyme E. Now, if you think about it, imagine you have an excess of substrate, so much substrate that you'll never run out of it, right? You can imagine how how fast A is converting to B, which refers to the rate of the reaction, how many molecules are converted per unit of time. It's like a speed. So how fast A is converted into B will be proportional to the amount of enzyme you have in there. There's actually a direct relationship if you have unlimited substrate. This graph shows you. So the rate, how fast many molecules are converted into product is proportional to the concentration of the enzyme. Just to give you an example, imagine you're working in a store and you have this huge lineup of people trying to come in. And you have 10 employees. You have thousands of people trying to come in. It's a super good Friday, it's called a Black Friday. Because if you have 10 employees, you can only serve X amount of customers per hour. Now imagine management say, well, we're gonna double. You have 20 employees. Obviously, you can go faster, assuming the door is big enough to hold all these people. Then say, okay, let's get another 30 employees. There are 30 now people, associates working there. You can go even faster. You can see how if you have unlimited substrate, unlimited clients, now how fast you can serve these clients, how much you can convert these substrate into product is proportional to the amount of enzymes, which will be the employee working in the store. Now, this is different if you have a certain amount of enzymes and different concentration of substrate. Imagine now you have, in the same store, you have 20 employees. And you start early in the morning, you have only 5 customers up, right? Slowly coming in. Obviously, you can serve them at a certain rate, which is determined by the number of customer or substrate. Now imagine it's 10 o'clock, you start having 20 people per hour. Obviously, how fast you serve them will go up. You still have enough employee to work on that. Then you start having at 11 o'clock, 30 customers coming per hour. And then at a certain point, whoa, after work, you start having 100 of them. You can find yourself in a situations where you don't have enough employees. You don't have enough enzymes to serve all these customers, to convert all this substrate. Right? And so at that point, you will reach some sort of saturation. Same thing with the enzyme reactions. You can see at the beginning, if you have very little substrate, the rate increases slowly, it will go up proportional to the amount of substrate you have. It will be linear. At a certain point, you will reach a point where you don't have enzymes. All the employees are saturated with work, right? You gotta hold on to some substrate outside of the door. Same thing here in the enzyme. All the enzymes are saturated with the substrate, and you reach a maximum speed in the reactions. Now, why are these two factors important? Well, you can see how cells can use, for example, the amount of enzyme expressed. You can have more enzyme expressed to make more product, or produce less and you have less product form. You can control how fast the rate of the reaction by just expressing more or less enzyme. Remember, for example, desaturases. We talk about those enzymes that convert the saturated fatty acid into unsaturated fatty acid, and how they are expressed depending on the temperature. That's exactly an example. You are at a high temperature, very little desaturases. Cold temperature, more desaturases is expressed. So you can control the conversions of saturated fatty acid into unsaturated fatty acid based on the need that you have in there. Same thing with the substrate. You can bring in more substrate or less substrate and control overall the rate of the reaction. Together with this, now, in living organisms, there are also a lot of molecules that are used to actually turn on the enzyme by acting directly on the enzyme molecules. Now, in general, these molecules, they are referred to as enzyme regulators, and they can work in two different ways. And so they can be referred to as competitive or non-competitive. And this depends on whether they bind to the active site, the same site where the substrate binds. In that case, they will call competitive, or in a different site. There are other sites outside of the active site where they could bind. These are referred to as allosteric sites. In this case, they're referred to as non-competitive are referred as competitive in this case because they compete with the substrate for the same site, the active site, and non-competitive because in this case they don't compete with the substrate for the active site. Now you can imagine how competitive regulators can only be inhibitors, because if they bind to the active site, the substrate cannot bind to it, so they block the reaction. Whereas molecules that bind to the different site than the active site, the allosteric site, instead can be either inhibitors, they can have a negative effect on the active site, or a positive side effect, on the active site. And in that case, that would be called allosteric activator. We'll talk about that in a second. Starting with the competitive inhibitors. So those molecules bind to the active site and they inhibit the activity of the enzyme. Now, these molecules, again, they will bind to the active site. Imagine this red molecule being the competitive inhibitor, bind to the same active site that the substrate will bind to. By doing that, they block the activity of the enzyme. They inhibit the activity of the enzyme. Now, they can do it in two different ways. They can either bind to this in a weak way, and so they can be actually displaced by the substrate. They can bind and be released, so the substrate can bind sometimes. They still slow down the reaction. In that case, they're referred to as reversible competitive inhibitors. Or they can bind and stay there forever. They bind so tightly that now they're permanently attached to the active site and therefore permanently inhibit the activity of the enzyme. In this case, they're referred to as irreversible competitive inhibitors. Here you have an example, very famous molecule, penicillin. Is the first antibiotics discovered by Alexander Fleming in 1928. So this is a competitive inhibitor of this enzyme called transpeptidase. This is an enzyme that is involved in the production of this peptidoglycan. Don't worry, you don't need to remember that. But it's involved in this important molecule for the cell wall in bacteria. So if you blocked the activity of this enzyme, you actually block or alter the formation of the cell wall in bacteria, leading to their death. That's how penicillin works. Now, if you look, this is penicillin, and this is the normal substrate of transpeptidase. I hope you can see the similarity. Look at this part of the molecule of the substrate here. You can see it's very similar to penicillin. In fact, penicillin basically simulates the substrate. So the transpeptidase will bind to this molecule or penicillin the same way. And by doing that, once it binds penicillin, penicillin is bound to the active site. It works as a competitive inhibitor. By doing that, obviously it will block the natural activity of transpeptidase. Now, as we said before, so that was competitive inhibitors. Okay, can be reversible or irreversible, and they bind to the active site. Now, there are other molecules, other regulators that can bind areas different than the active site. They're referred to as allosteric regulators. So imagine this being an enzyme. Now, this enzyme would have its active site here, that's where it binds, and then would have a different site referred to as the allosteric site. This is the regulatory site. It can be bound by different type of molecules. So now, if it's bound by molecules that basically increase the activity of the enzyme, it will be bound by what is called an allosteric activator. If instead the binding here results in the inhibition of the active site, at that point you would talk about an allosteric inhibitor. Okay. Now, how is this done? Basically, the effect of the molecule binding to an allosteric site, it basically has an effect on the activity of or affinity state of the active site. So the active site in any enzyme can be in a high affinity state or low affinity state. So in a high affinity state, you bind the substrate very well. In the low affinity state, it wouldn't bind the substrate very well, or it wouldn't bind it at all. So these molecules, the allosteric regulator, bring the enzyme either in high affinity state. If they bring it in high affinity state, there would be allosteric activators. If they bring it instead in low affinity state, there will be allosteric inhibitors. Okay. Now, an allosteric inhibitor, again, will bind to the allosteric site. And when it does that, it will bring the enzyme that before the binding will be on high affinity state. So will be binding the substrate, making the product. Now, once it binds the allosteric inhibitor to the allosteric site, now this enzyme will be turned into low affinity state. And by doing that, the substrate will be released, and no other substrate could bind or could bind with very much less efficiency. So the binding of this regulator, allosteric regulator, has resulted in the inhibition of the enzyme. So it's called an allosteric inhibitor. So not surprisingly, many times these allosteric inhibitors, so this molecule here, that block the activity of an enzyme, are actually product themselves of reactions of the same enzyme or other enzymes further down on a pathway. So why would it be? Imagine this in the enzyme that converts this substrate into the same product here. So you can imagine how this will work very well when the concentration of the product is very low. And the more you do it, the higher it becomes concentration, and the higher is the chance of this molecule binding to the enzyme and stopping the reaction. By doing that, the enzyme, the product of the enzyme, would actually control how much is produced of itself. At low concentration keeps the reaction going. At higher concentration, it starts the inhibition effect. So the binding is proportional to the concentration. Sometimes, like in this case, this is the pathway through which this amino acid, isoleucine, is produced starting from threonine, another amino acid. This consists of 5 reactions. After each reaction, so it's a pathway, all these reactions are connected with each other. So the product of the first reaction here is the substrate of the second, product of the second, the third, and so on. Now, you don't need to remember this reaction, by the way, but you have to understand the point. As you can see here, now, isoleucine itself, it works as an allosteric inhibitor of the first enzyme, not surprisingly. So what it means is that once this pathway has been going on for a certain time, you start having a lot of isoleucine being produced. And you want to stop that. And by doing that, isoleucine itself will bind to the allosteric site of the first enzyme, the deaminase. And by doing that, you prevent the formation of more isoleucine. Notice that it's important that it binds to the first enzyme, because there is no point of binding to another enzyme further down in the pathway and accumulating a lot of intermediate. There will be a lot of loss of energy here in the meantime. Okay, so oftentimes, allosteric inhibitors are actually products of reactions themselves that do control through a process referred to as feedback inhibition. They do control how much of themselves ultimately are produced, either by a single reaction or by a pathway, like in this case. Allosteric activators that work in the opposite way. They bind still to the allosteric site, okay? But in this case, what they do, the effect that they do is they bind to the enzyme that it was first in a low affinity state, so it was incapable of binding the substrate. After the bindings that they bring it to a high affinity state, you can imagine how all of this is done through effect to the secondary and tertiary structure of the molecule, which then affects the active site, which now is a high affinity state. It can start binding the substrate, so has been turned on, hence the name allosteric activators. Together with this mechanism, now enzyme can also be activated through covalent modification. And one example is the one we went through when we were talking about signal transduction. If you remember, after a signal molecule is bound by a receptor, then you have the signal transduction taking place inside a cell. And this is mediated by kinases. Kinases are enzymes that actually modify other enzymes, other kinases, by trying a phosphate group covalently to the target molecule. By doing that, they actually turn on an enzyme, the target here. It gets turned on, right? The opposite is what is done by phosphatases. They do remove this phosphate group, and by doing that, they bring the molecule back to the inactive state. That's an example of regulation of enzyme by covalent modification. Many cells do that, as you know, during signal transduction, and plus there are other molecules that can be added to achieve that. Finally, the last two factors that we'll talk about here are temperature and pH. How do they affect the activity of an enzyme? So both of them have a big effect on enzyme, and every enzyme would have an optimal temperature and pH where they work. And then when you change that, there will be a big change in the activity of the enzyme. So pH first. Here you see on this graph right here how this is the curve of the enzyme activity. So how fast is the reaction done by these enzymes at different pHs. So these are the pH going from 0 all the way to 11 in this case. You can see this is pepsin. This is an enzyme that works in your stomach. It works very well at low pH, has maximal activity. So the reaction that it does is the fastest at a pH of 2, very acidic pH. But then it gets turned off, particularly when you go to high pH. So it's active in your stomach and inactive in your intestine, where you have a pH of 7. So it gets inactivated. Other enzymes, that most of them work very well instead in the range of 7. You can see here different trypsin and so on, other enzymes important for digestion and so on. So how does pH affect that? Well, you can see basically pH, if you remember, is proportional to the concentration of proteins that there are around. And so if you imagine, if the more you have of protons around, the more you change the charge also in the proteins. So imagine this group here. This is positively charged. This will be negatively charged. The more protons you have, the more neutral this will become. Hence, the interaction between these R groups will be affected by that. And this will be felt in the rest of the structure, the same thing here. So the way pH effect is basically mostly affecting ionic bonds inside the proteins. By doing that, they affect the tertiary, and eventually quaternary structure of the protein, and also the structure of the active site by increasing or decreasing the activity of the enzyme. Similarly, temperature. You can imagine how by heating up a protein or cooling it down, you alter ultimately the amount of kinetic energy present inside the protein. So these are groups that form the tertiary, eventually quaternary structure, and even the secondary structure with the hydrogen bond is very sensitive to the amount of kinetic energy that is present there. If you heat it up, there's so much energy that you can break these bonds. That's the process of denaturation. Or if you cool it down, this bond becomes too rigid. So the molecule can't even move around. The active site cannot do the induced fit anymore. Right? So you can see how temperature can affect ultimately the amount of kinetic energy in the enzyme, and therefore the secondary, tertiary, and quaternary structure eventually of the enzyme. And you can see it when you look, you study the activity you're doing BIO359. If you look how fast an enzyme can perform a reaction at different temperature, you see that it has an ideal temperature. Most of our enzymes are in the range of 37 degrees, not surprisingly. And then if you cool it down, the rate of the reaction will go lower. If you go too hot, it would actually go down much faster because at a certain point you start also denaturing the enzyme itself. So this enzyme can be also turned on even in living organisms with temperature. Imagine some enzymes involved in fever events, right? They have to be more active at a higher temperature than lower temperature. So there are another mechanism through which enzymes can be regulated. So in summary, what did we talk about here? So we talked first of all, why do we need enzymes? We need them to speed up the reaction and to have control over the reaction. And we talked about how they work. So how they affect the activation energy of a reaction. They don't change the delta G, remember. And how do they do that? By binding the substrate, either by bringing them together or altering their state. Now we talked about the regulatory mechanism of enzymes. We talked about the concentration of enzyme and substrate affect how fast a reaction, an enzymatic reaction goes, so affecting ultimately the reaction itself. And we talked about how some molecules called inhibitors or activators can bind to enzymes, either on the active site, in this case you would talk about competitive inhibitors, or different site called the allosteric site. By doing that, they can inhibit or activate enzymes. And finally, we talked about how some enzyme can modify covalently by attaching some groups, such as the phosphate group, and the effect of pH and temperature on the activity of the enzymes as well.